All right. Good afternoon, everyone. Welcome back. And we will be finishing up lecture six today. It's a particularly long lecture. We had two classes on it already, and hopefully we can finish it with the third today. So picking up where we left off. We were talking about the purpose of cell division and the need to replace lost or damaged cells, growth and development, and some organisms use cell division as a way of asexual reproduction. And before the cell divides, before the cell splits into two, it needs to copy all of its DNA. And I just want to take a moment to refresh your memory on what DNA looks like and how it's packaged in a cell. The double helix that we learned is basically we call it the naked DNA. It's just nucleic acid on its own. It's not wound up against any protein or anything like that. So in that regard, it is in its most fundamental form, if you would, and that doesn't really exist inside a cell because we are not going to be able to pack all these DNA into the nucleus, right? And a lot of times the DNA is going to wrap themselves around these histone proteins. Previously, I told you they are like the yo-yo. You take the rope, which represents DNA, and you wound them around the yo-yo core, right? And that allows you to package them nicely. And once you package them a couple of times, then eventually you're going to get to a form called chromatin. And these are DNA wrapped around histone proteins. That's the form that you will find DNA in within the nucleus. So when the cell is not dividing, when the cell is not moving chromosomes around, you will find the DNA in this form, okay, chromatin. When you have to split the cell, when the cell is actually undergoing cell division or mitosis, then you need to compact the DNA even further to make it easier to move around and manipulate them. And so the chromatin will package themselves around more proteins and it forms these supercoiled and condensed version called the chromosome. Okay. So chromosome, chromatin, DNA are all made up of DNA. It's just a different level of condensation. Okay. The most compact, the most condensed version being the chromosome. All right. Now, in humans, we have a total of 23 pairs of chromosomes, 23 from mom and 23 from dad, which makes up a total of 46 chromosomes. Okay. Of the 46 chromosomes, 44 of them, or 22 pairs of them, are called autosomal chromosomes. Autosomal chromosomes, or autosomes for short. These autosomes basically determine all aspects of our body except for the sex. Right? So your eye color, your height to a certain extent, your blood type, your whether you can curl your tongue or not, whether your earlobe is attached or detached, the kind of hairline you have, curly hair, straight hair, all these features about us, they are all encoded within the first 22 pairs of chromosomes. And we call these autosomes. The sex is determined by the sex chromosome. If it's XX, then it's a female. If it's XY, then it is going to be a male. And since The male has the Y chromosome. The father is responsible for determining the sex of the baby. If the sperm pass on the X chromosome in the dad, then it's going to be a daughter. If the sperm pass on the Y chromosome, then it's going to be a son. Okay, so first 44 chromosomes, it's everything about you except for the sex. And then the last pair, number 45 and 46, that would determine the sex of the individual. When it comes to the number of chromosomes in an organism, there's no relationship between the size and the number, right? Humans have 46, but something much smaller, like a goldfish, can have more chromosome than us. Okay, and in some cases, for really really big plants, for a lot of plants, they have a really big genome. They have a lot of chromosomes. Okay? Things like this fern has up to thousands of chromosomes. Yeah, so there's no correlation between the size of the organism and the number of chromosomes, and there are also no correlation between the complexity of the organism and the number of chromosome it has. All right, something that's like a some kind of deer-looking thing right here. It's called mugec, and it's bigger than human, but it has only 6 chromosomes. Okay, so I want to take a quick note with you. This is quite short. It is called the cell cycle. And I want to draw a big circle here. This is the cell cycle. So we start with a cell here, with the nucleus, and then it's going through the cell cycle. The cell cycle, it's divided into two main events. Right? So this part right here, this 90% of the time, the cell is going to be in this part of the cell cycle. And this part of the cell cycle is called interphase. Interphase. It's just the yellow part, okay? And that's approximately 90% of the time the cell is going to be in this interphase. And so what does it do in the interphase? Well, in the interphase, it is performing, the cell performs normal function. Whatever the function is. If it's a muscle cell, it's going to be contracting. If it's a liver cell, it might be detoxifying the blood, so on and so forth, right? Whatever its normal function is doing it in the interphase. They will also be growing, gathering nutrients, resources, right? And generally preparing for cell division. Now, how long is it gonna take this whole cycle? It depends on the kind of cell. Some cells divide pretty rapidly, like your skin cell, they get replaced, every couple of weeks. Um, Other cells take longer time to divide. Some cells don't divide at all, right? Like your brain cells, they don't divide, right? Your heart cells, they don't divide. Um, so it depends. Over here, this small part, that is the actual cell division, and that's mitosis. If the cell divide, it's gonna take about 10% of its time in the cell cycle to do the actual division. And with the successful division, you will get two cells, and each of the cell will be able to go through another round of the cell cycle. Yeah, so this is we call it daughter cell. Each daughter cell will go through another round of the cell cycle. Now, within the interface, we can further divide it up into different stages. Okay, so we have what's called the G1, and mostly it's just growing. So growth, and then we have the S phase, where you will be doing DNA replication. You know, all those stuff that we learned last week, the helicase, the gyrase, and all that stuff to copy the DNA. That happens during this S phase. S stands for synthesis. And then you have G2, which is more growth. But along the way, and this is very important to understand because the lesson is going to come back to this point again and again. Between G1 and S, there is something called a G1 checkpoint. So there's like a checklist that the cell needs to go through before it allows to move on to the next phase of the cell cycle. The next phase being DNA replication. So there are a couple of things we need to check. So for example, we need to check the size and volume of the cell. Like, is it big enough to divide? If this cell just formed from a recent division, maybe it hasn't had time to grow big enough yet. Then we don't have enough cytoplasm to divide. So, you shouldn't divide yet. You should grow a little bit more. Right. Another thing that you need to check would be check the DNA quality. Example, what are you checking for? Damage, air, telomere length. We talked about telomere last class. If they are critically short, the cell is not allowed to divide again. Because every time we copy our DNA, the telomeres get shorter. If the length is already critically short and then you go ahead and you copy it, it might get damaged. The actual DNA might get damaged. You should also check for resource availability. Do you have enough ATP? Do you have enough nucleotides to build the new DNA? We're talking about billions of base pair. Do you have enough of those nucleotides floating around? All right, so you got to check all these things. If the answer is yes, like everything is okay, no damage, no air, so on and so forth, then the cell is going to be allowed to move on to the next part. If it is not okay, okay, so if checkpoint fails, then cell enters growth arrest. Okay, is not allowed to divide. Okay. It might becomes senescent, right? Like this old cell, right? Not permitted to divide anymore. Or undergo, what do we call it when the cell kills itself? If it has irreparable damage, right? DNA is too damaged. There's no way to fix it. The cell we shouldn't keep this around. We should kill it. What's the word for it? Some of you must remember.
Autophage.
Uh oh, I think
Phagocytosis.
yeah, autophagy is another thing. It's like the cell eats itself, but I don't that's not the word we use though. Um,
Phagocytosis.
it's not the one we're looking for. Thank you though.
Killed by
Anyone
the
else?
lysosome, like
Yeah, that's right. It involves a lysosome. Anyone else want to try before I tell it? That's right, Samuel. Thank you. Apoptosis. Apoptosis, which is programmed cell death, right? There we go. Right, so like sometimes it's dangerous to keep these cells around, especially if it has so much airs accumulated, right? Like we don't want it to mutate further, so kill it off, right? But only if the checkpoint fails. Right? If it does not fail, then we can go ahead and copy the DNA. Just gonna move this over here. Sorry. And then we're gonna have a second checkpoint right here between S and G2. This is called a G2 checkpoint. I need you to understand the importance of these checkpoints. When we talk about cancer in a bit, it has to do with these checkpoints, okay? So G2 checkpoint, uh, by this point, you've already copied all your DNA and you want to check the quality of duplicated DNA. And all the DNA that you just copied, you gotta make sure that you didn't make any mistakes. Did you copy everything? Did you copy everything correctly? Right, so on and so forth, right? And sometimes you make mistakes. DNA polymerase 3 and 1, they have spell checking mechanism. They can fix the error, but even so, it's possible to still make a mistake. So you basically do like a final check here before you proceed. Right? It's like, if you type a 10-page essay, right, before you submit it, you're going to proofread it, right? You're going to ask people to look over it for you, right? That's basically what G2 is. All right. So it's also possible that you make so many mistakes during the copying process that is not repairable, or like something went wrong during the DNA duplication process, and and you cannot fix the error, then the cell will also exit the cell cycle. Depending on the severity of the situation, it might undergo growth arrest, become senescent, or you have to kill it outright. So to go through the cell cycle, you have to pass G1 and G2. Right? It's like getting the driver's license in Ontario. Right? You gotta pass G1, G2 before you can go forward. Right? Otherwise, you have to exit the cell cycle, and it's gonna be one of these three things. All right. So basically, a cell can go through the cell cycle quickly. If it goes through cell cycle quickly, then it's gonna divide, divide, divide. Sometimes it doesn't go as quickly. Then you don't have as rapid of a division. Now, eventually, if all goes well, then you're going to be allowed to go to actual cell division, the mitosis step. And this mitosis step has 4 steps in it, but we're going to do it later. Right? For now, you just have to know mitosis is the actual cell division. So I just want to pause for a moment and see if you have any questions. Okay. Um, Here are the PowerPoint slides for it. Interface, the cell is performing its normal functions. It's growing, it's preparing for mitosis. And then the mitosis is the actual cell division that takes one parent cell and then it splits and become two daughter cells. Yeah, we use feminine description for these things. And there's no like son cells or anything like they all call daughter cells. This is basically the cell cycle I've drawn for you. Um, Again, there are various checkpoints. There's another checkpoint here they didn't put here. But if it fails, then it's gonna exit the cell cycle. Okay. Remain specialized means it stays the way it is. Growth arrest. It's not allowed to, you know, divide any further. Okay. So we mentioned about the checkpoints already, and what happens if it fails the checkpoints. We're gonna come back to these checkpoints later. You might wonder, like, what's actually doing the checking, right? Like, what kind of mechanisms are in place to detect these errors? We're gonna get to it in a bit. But for now, let's go through the steps in metaphase. In mitosis. So this is interphase. This is not mitosis yet. This is just when the cell it's um, going through the G1, the S, and the G2. And we have a cartoon picture over here, and at the top we have a real picture. So in reality, the cell are there's no color, right? They are all colorless. They're so thin, light just passed through them. You are seeing color because people who prepare for these slides, they can stain them, they can dye the cells certain colors. And there are only three colors you can see here, and I won't tell you what they represent. Okay, so when you see the blue in the real picture here, the blue is where the nucleus plus chromosomes is going to be. And right now, you don't really see the chromosomes because the chromosome hasn't formed yet. It's still interphase, so they are still in the form of chromatin. And chromatin is not very visible because they're not very condensed yet. The red is just the general location of the cell of the cytoplasm, if you would. And then the greenish, yellowish stuff, that's the spindle fibers. We learned about spindle fibers in lecture 3, I believe. These are basically cytoskeleton that are created by these centrioles, and the spindle fibers, they will help guide chromosome movements. All right. So what's going on in interphase? Nothing much. Right? Like we said, the cell spends most of its life in this stage, about 90% of the time, performs regular function. But, along the way, it's going to go through the checkpoints, and if it's good, it will copy its DNA. Hold on, let me see what's going on with my slides. Um, okay. I got to show you this thing quickly. You should copy this down too. All right, so let's say we look at a chromosome, right? Doesn't matter. I'm just gonna pick a random color here. Chromosome. There's a chromosome. All right, so this is chromosome. Let's say this is chromosome 1. Okay, so you will have chromosome 1 from mom and chromosome 1 from dad. It doesn't really matter which one is from in this case. So what happens is when you have DNA replication, you will make an exact copy of this thing. So instead of just one, now you will have another one that looks exactly the same, and they will be stuck together at a point. Now I want you to think about this. Okay? How many chromosomes do you see over here? That's a rhetorical question, right? You will say one, right? And that's correct. That's one chromosome. How many chromosomes over here after DNA replication? Give me some answer.
Two.
Two, yeah. That's what most people would think, right? That there are two chromosomes now, right? Like one and two, right? But the way we count chromosome is like this, right? If they are stuck together, we count it as one. Right? If they're connected, it doesn't matter you have two pieces, we count it as one. Right? It's like Kit Kat, the chocolate bar that you can break into half, right? Like before you break it, we still count it as one. Okay? It's only after you break it apart that, oh, you have two pieces, right? Okay. So over here, this is still considered as, count as one chromosome because they are stuck together. Okay. And these are exact copy of each other. So we have a name for it. We call them sister, sister chromatids. Again, like it's feminine, the word that we use to describe these things. Sister chromatids. So these are identical. Now, we learn a lot of C words, right? So don't confuse chromatid. This is TID. Do not confuse that with chromatin, TIN. There's only one letter difference. Chromatin is the form of DNA that's wrapped around the proteins. That's what they look like when they're in the nucleus before you copy them. After you copy them, then you have the sister chromatids. Okay, they are stuck together. Sister sticks together. Um, that's what I tell my girls, right? You two are sister, you have to stick together and help each other out. So these sister chromatids are stuck together, and the location at which is stuck together, it's called the centromere. Centromere, right? Connecting point between sister chromatids. All right. Now, just I know it's already confusing, perhaps, but I just want to throw in an extra term here for you. What are these pink stuff that I just shaded? What what's the name for that?
Telomere.
Thank
Telomere.
you. That is absolutely correct. Telomeres, right? They are located at the ends of the chromosomes, right? Ends of the chromosomes. Oops, totally messed it up. Okay, so centromere, telomere, sister chromatid, and this whole thing is a chromosome. Okay. So this is where it sometimes it gets confusing for people, and I understand why. This is the parent cell. Okay, one more time. How many chromosomes do we have in a parent cell? How many chromosomes in a parent cell? Yeah, 46 is correct. When I say parent cell, I do not mean like our parents. Okay. In our body, if a cell is about to divide, that cell is called a parent cell. So Before the cell divides, it has to go through G1 and then checkpoint. And then if it's good, then you do DNA replication. So each of the 46 chromosomes will get copied. And now, after you copy them, how many chromosomes will you have? Think about it. Okay? Please be careful. How many chromosomes will we have now? Tell me. Anybody? Any brave soul? How many chromosomes do we have now? Okay, we have 46, we have 92. Anyone else? 92. Why don't we do a vote, okay? I want to know. I want to know what you guys think. Okay. If you think is still 46, choose A. If you think is 92, choose B. If you think is 23, choose C. If you have other numbers in mind, choose D. All right, pick something, guys.
It is like 92, but we count as one.
So which means is what's the final answer?
46. Then
That's right, still 46, right? Okay, the answer is still 46. Why? Okay, let's see. Let's not use 46 because I cannot draw 46. It's gonna take forever. Okay, you don't have to draw this, you can just watch, okay? So let's say we have 10 chromosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 chromosomes. They're not gonna be the same size in reality, but doesn't matter, okay? Now, what happens is I'm gonna copy all of them now. I'm gonna copy all of them. Okay, and when you copy them, they are stuck together. They are stuck together. Sister chromatids are stuck together at the centromere. Okay, so each one is gonna get a clone of itself and it's gonna be stuck together. So if we have 10 chromosomes before, after we copy them, how many do we have now? Still 10, right?
Yes.
Because they are count that way. Is that okay?
Yes.
Thank you. Um, like I said, hold on. Let me show you the chocolate bar. I don't know if do you guys know this chocolate bar? The Kit Kat, right? It comes in, you can break it into two pieces, right? So like when it's together, it's count as one. When my kids were younger, like I can fool them if they want if they want the Kit Kat bar, and then it's just like I want two, right? And then I would take the bar and then break it in half. And now I give you two. It's still actually just one bar, but then once you break it, you count it differently. They don't fall for that anymore, but it's the same concept. All right. So if you start with 46 chromosome after DNA replication, it's still 46. Then we will see afterwards that we are gonna separate the cells, and then over here we will have 46 and 46 again. So this is called daughter cells. Excuse me, daughter cells. All right, so let me show you the PowerPoint slides again over here. Next, we're gonna proceed to prophase. I'm just go through the points with you first, and then we can draw it out to make it more clear. So this is early on in prophase, and if you focus on the picture here, the real picture, you can start to make out the outlines of the chromosomes, right? By the time you get to prophase, remember, you would have copied all your DNA already. So the chromatin has now become visible chromosome. You couldn't see it before. It's still very uncondensed relatively. And then in this early prophase, it's more visible. There's a question that asks if 46 becomes 47, that will be Down syndrome. Not necessarily. It depends which one is extra, right? If the extra chromosome is 21, then it's Down syndrome. It could be extra 18, then it would be trisomy 18, which is another syndrome. We're going to talk about that later on, though. Okay, good question. DNA replication is completed. Sister chromatids are held together at centromere. We talked about that already. Now we got to get these chromosomes out of the nucleus. And as you know, the nucleus is surrounded by the nuclear envelope. So we need to take away the nuclear envelope to allow the chromosomes to come out. Right? If they're all scrunched in the nucleus, it's really hard to move them around. The centrioles will produce these spindle fibers, which look like that um, as the centrioles move to the opposite side of the cell. If you look at this picture, you can see these two yellow dots in the center. Those are the centrioles. All right. We're still in prophase, which is the first part of the metaphase. Okay. Um, right now, later on in the prophase, you can see the nuclear envelope is completely gone, and then you can see the individual chromosomes very clearly. Now, we have the centrioles here at the opposite end and producing the spindle fibers. All right. Now, I want to draw this with you together to, usually it's better when you draw it out. I mean, if you took biology before, you probably know this already. But if this is your first time taking it, drawing it out is going to help you. Okay, so this is the prophase. Okay, prophase. Okay, so for mitosis, um, you can remember there are four steps. Okay, PMAT, P-M-A-T. First step is prophase. Again, you are not going to be at this point unless you have passed all the checkpoints, right? You've gone through G1, you've copied your DNA, you've gone through G2, everything checks out, you're good to divide. Now you're going to be doing the prophase. So what's going to be happening is for prophase, once again, we're going to have these organelles. We learned these organelles in lecture 3. They are called centrioles, and they produce these spindle fibers. Now, for simplicity's sake, I am only going to be tracking two chromosomes, okay, two pairs of chromosomes. So let's say we have chromosome 1 from mom and chromosome 2 from mom. Of course, there are 3, 4, 5, 6, 7, 8, 9, 10, all the way to 23, okay? And then we have chromosome 1 from dad, which would be the same size as chromosome 1 from mom, and then chromosome 2 from dad. But we are at prophase now, so each of these chromosomes would have been duplicated. They would have been copied, and they would have been held together at the centromere. Okay, so let me just give you a little bit of description here. This would be a duplicated chromosome. Okay, now if the numbers help, put it in. If it confuses you, don't. This is chromosome 1 from mom, and now you have two copies of it. Chromosome 2 from mom, and now you have two copies of it. Chromosome 1 from dad, two copies. Chromosome 2 from dad, two copies. Right? Before DNA replication, it's only one from mom and one from dad, right? But now at this point of the game, you have two from mom and two from dad for each chromosome because you have copied them. But nevertheless, they are still stuck together. Okay, so at this point, it is still 46 chromosome in total. But, I've only drawn 4. so you don't get confused. Okay, if it helps, I really don't know if this helps. Sometimes it helps, sometimes it confuses the heck out of people. You can say 92 sister chromatids. 92 chromatids, if you would. Okay. Only if it helps you. If the 92 confuses you, don't do it. Okay? Just do 46 chromosomes. Okay, so nuclear envelope is gone, right? I'm gonna pause a little bit. Anything you guys want to ask at all? Okay, so there's a question. It says mitosis is asexual. So what is that chromosome? Why is that chromosome here? Yeah, okay. So asexual reproduction means when an organism just split and become its own offsprings. Okay, so no partner is required. So we talked about starfish last time. We talked about those amoebas. They're able to split. So in those organisms, we would not classify the chromosome as, mother chromosome or father chromosome. But you got to understand, mitosis is not only used for asexual reproduction. We do mitosis in our body for other purposes, not related to reproduction. All our cells, except for like the brain cells and the heart cells and a few um, exceptions, our cells are able to divide so that we can grow, so that we can develop, right? So let's say I have a skin cell here. In that skin cell, we would have the maternal and the paternal chromosomes. Okay. So that's what we're talking about. This is like a cell in our body. Okay, let's keep drawing and see what happens. After prophase, we're going to have metaphase. Metaphase. Now, to help you remember it, you can think of P. P is for like preparing. You're preparing to move the chromosomes. Yeah, I don't have space here. Okay, you can put prepare. Okay, prepare. M is for middle. All right, so in this case, the centrioles, which are these little potato tots looking things, they will send the spindle fibers out, and the spindle fibers will attach to the centromere of each chromosome. Okay, they will attach to the centromere of each chromosome. And then they will pull them back and forth. Okay, some of them might not attach to anything. Again, that's okay. Alright, so they attach to the centromere here. The spindle fibers will pull on both sides. It's like a tug of war. You guys know tug of war, like the rope, and then you have two teams of people like pulling on each side. Okay? That's basically what's happening here. The chromosomes get pulled one way and then another, and eventually they get lined up perfectly in the middle of the cell. Okay, and that's what metaphase is for. Okay, spindle fibers help line up the chromosomes at the center of the cell. Okay, that's metaphase. Next, we have anaphase. A for apart. Pulling the chromosomes apart. Oh, by the way, over here, it is still 46 chromosomes, right? 46 chromosomes. Next, I'm going to draw another one here. The spindle fibers will continue to pull so hard that the sister chromatids, these two things, will separate. They will separate, and then it will get pulled towards the opposite side of the cell. Okay, so sister chromatids are separated and get pulled towards opposite ends. So I want to ask you again, how many chromosomes are we talking about now in this at this point? How many? We started with 46.
92.
92 is correct because now they are separate and we have to count them separately. So now it's 92. 92 chromosomes. Okay. I hope it makes sense. And finally, we have telophase. Telophase plus cytokinesis. That's a big word. Cytokinesis. Okay. T is for tearing. Tearing. Okay. So basically what's happening in telophase is the opposite of prophase. The opposite of prophase. So the centrioles are going to be here now. Okay. Our nuclear envelope is going to start to form again. And then the chromosomes, look, listen to me for a second, right? Like this chromosome is exactly same as that one. This one is exactly same as that one. So when you separate it, each half of the cell will get exactly what you started with. And the chromosome will now uncondense back into chromatin. Okay, the mother chromosome will become the chromatin again, and then you have the father one. Okay, and then the cell here is basically gonna cleave itself and cut it, and then you get two daughter cells. And that's all. That's all there is to it. Right? Cytokinesis, this word, like what does it mean? Forgot to tell you. This is the division of the cytoplasm. Division of the cytoplasm. All right. That's it.
I have a question.
Mhm.
End of this process, how many chromosomes we have for each?
Yeah, so like it's 92 here now. If you think
Yeah.
about it, this is gonna split. So 46 for one and 46 for the other, right?
Okay. Yeah. Okay.
Which is the whole point of the situation here. You start with 46 and you end up with 46 in each. Okay, so hopefully that makes sense. You can look at the slides. It's exactly what we talked about. This is onion cell, guys. I want you to spend a moment and see how many of the phase in mitosis you can identify. Okay, like I mean A, B, C, D, take a moment and try to figure out yourself. I'll tell you the answer in a bit. All right. Anybody? Let's start with something easy. What do you think this one is? Actually, A is pretty easy. I don't know. Like I know the answer, maybe that's why it's easy. But anybody want to share what A is? What do you think this is?
Um, metaphase?
Profes,
Yep, this is
profes.
metaphase actually, because it's all in the middle. Okay, this is the whole cell, and you can see they are all in the middle of the cell, right? If it's prophase, it would still be kind of, still kind of in the nucleus a little bit. Nucleus is starting to dissolve, and you know, um, maybe it'll be more clear in another one. How about this? Does anybody want to tell me what that might be?
Anaphase.
Yes, that is anaphase because the chromosomes are starting to move apart. Okay, there is an other anaphase right here. Okay, do you guys see an other anaphase maybe?
Yeah. Uh,
Wh- Which
the
row?
last
Yeah,
last
The
row.
last row.
at the bottom.
Yeah, there you
At the
go, right?
bottom.
Another anaphase, right? Okay, this one is telophase.
Telophase.
Yeah, telophase. And how do you tell telophase from anaphase? Telophase, the chromosomes are more clumped together, and you can see that the middle is starting to divide, right? And these things are starting to form back into the nucleus, right? So that's how you can tell. Whereas anaphase is still, you can still see the individual chromosomes.
More
Okay,
condensed. Yeah.
yeah. And D over here, that would be prophase. Prophase. Okay, these guys, they're I would say these are not mitosis. They're not dividing yet. They are still in their interphase. How do I know? Because I can't see the chromosomes. Chromosomes are still, you know, in the form of chromatin. You know? So be prepared to identify some of these pictures on the test. I'm not going to give you something like really weird like that. Like that's hard to tell. Okay. If I'm going to ask you to identify it, it's going to be something that's straightforward. Okay. I'm not going to do the polls. You guys can do it later. Oh, maybe I'll do this one. This one is important. Go ahead. Yeah. Thank you for those of you who picked something. The correct answer is 16, right? For the reasons that we've talked about. Once the chromosomes separate, then then you count them separately. Yeah, that's okay. If you can't see the poll, it's fine. You can do it in your own head, okay? And check the answer. This one is just like communication type of question. We don't have time to take it up. You can think about it. If you if you have an answer, feel free to send it to me through email, and then I can take a look at it. Okay. All right. Before we talk about the checkpoints, since I have most of you here, I just want to bring your attention to something. Um, I told you last time that we are changing the way that we're doing the tests, that you have to turn on the camera and stuff like that. I've received a few questions from students, and so I actually posted a new announcement. 17 of you already read it. If you haven't, please go ahead and read that. But you can also find the same information under course information and assessment policies. Okay, please pay attention. This part is important. For the next quiz, which I forgot when is going to happen. Um, I'll take a look in a second, okay? For the next quiz, you will have to do some rough work to answer some of the questions, perhaps, okay? It's not something that you can just answer directly from your head. And so, I mean, if you can answer directly from your head, fine, no problem. But if you need to do some rough work and you need to write down something, this part is important, okay? First of all, if you're gonna bring paper, make sure that the paper doesn't have anything on it, okay? It should be blank. And when you are doing the question, when you're doing some rough work because trying to figure out the questions, when you're writing things, you need to tilt your camera so that we can see your hand and that you are writing stuff. Okay? And then when you're ready to answer, you can tilt back. Now, when you tilt your camera, it's possible that the program will say, "Oh, we can't see your face. You should show your face again." That's okay. You can just keep on doing your work until you're ready to answer, and then you can show your face again. Does that make sense? At the end of the test, of the quiz or test, whichever one you're doing, if you have done rough work, then you have to take pictures of the rough work at the camera. At the end of the test, it will ask you to submit your rough work. Okay, and then you will do that towards the end. If you didn't have any rough work, then you don't have to submit it. But if you did, you will have to submit it. Okay. Uh, it's a little bit different from math. I'm not gonna have like a separate folder for you to upload high quality pictures because I'm not marking your rough work. I just need to see what kind of rough work you did. That's all. All right. So just do it at the end of the test and it'll be fine. Yes, question.
Yeah, so teacher, in math, we have to put the computer far away because she wants to see everything surrounding and while you're
Yeah.
doing solving the question. Do you require something like that or
If you can do it, great. But then, I mean, like for math, you don't have to choose answers on the screen, right? My
yeah, yeah, yeah.
understanding. Yeah. For me, you have to. So like if your screen is really far away,
Yeah.
like it doesn't make sense to you to pull it back and forth, right? Like, you know what I mean? I
Yeah.
okay, so like here's the thing, guys. Like use common sense. Like for those of you who are being honest, who are not cheating or anything like that, then you don't have anything to worry about. Okay. It's like for people who are doing suspicious stuff and doing things that they're not supposed to, right? Then like you should stop doing those things. Okay. Use common sense. If you are writing this test in classroom in an in-person setting, and in those settings, if you're not allowed to do certain things, then you probably shouldn't be doing it on an online environment either. Okay? So like no phones, no tablets, no smartwatch, that kind of stuff. Okay? And also, now this is important, I forgot to mention. If if your laptop or your computer is connected to a second screen, please unplug that second screen before starting the test. Okay. We don't want you to have additional monitors while you are doing it. Okay. Also, for this upcoming quiz and the final test, there is something like it's not a periodic table, but it's like a periodic table. Like you need to use it to do some of the questions. So I'll be providing that for you in a digital format. Okay. I'll show you how to do it when we get closer to the test day. So just read through these things. It's very important that you understand what the rules are. Um, Let me see when the quiz is. I don't remember. Sorry, it's like too many classes going on. Can't keep track. It's next Wednesday. It looks like it's next Wednesday. We should be able to finish lecture 7 by then. We'll see. I'll double check. Okay, and then I'll let you know. Someone asked if they can do the test on campus. No, we have to separate the in-person from the online people. So, online is online, in-person is in-person. Okay. But like I said, there's like we're not trying to hunt down anyone, okay? So, don't get extra stressed out because of this. If you're just doing the test like you're supposed to, you don't have to worry about it, okay? Having said that, any questions before we move on? Okay. If you think of anything, feel free to email me. I will address your concern. Let's keep going for now. Well, actually, it's you guys need a break? Maybe. Okay, maybe I'll give you 5 minute break, okay? It's like has been an hour already. We'll come back. 1:30, 36. Right, and then we'll resume. See you in a bit. All right, let's continue. Now, this next part is very confusing. In spite of my best effort, sometimes people still don't fully understand. So please stop me if I'm not making sense. All right. Let's take a closer look at those checkpoints. Okay, the G1 and the G2 uh, checkpoints, right? So just to quick recap, G1 is to check to see if all the qualities of a DNA is good before you go on to copy them, and then G2 is after you copy them, like did you do everything right, right? That's basically the checkpoint. Now, what's actually doing the checking, right? Like, do they have like mini peoples inside with clipboards and checking the boxes and kind of, okay? But instead of mini people, the ones that are doing the checkpoints are proteins. Right? So there are various proteins that are important for regulating the cell cycle. We can separate these proteins, these regulators, into two categories. They are either there to promote mitosis, we call them growth factors, or they are there to slow it down, we call them growth inhibitors. The best way to think about it is growth factors are basically proteins that acts as green lights. Okay? What do cars do when they see a green light? They move, right? And the inhibitor would be like red lights. They stop the cell cycle. Okay? So green light, cell cycle progress, the cell divides. Red lights, cell cycle stop or pause, and you do not divide. These are proteins, okay? Which means to make these proteins, we need the instruction from DNA. And since we know DNA can contain mistakes, DNA can contain mutations that would cause the instruction for making the proteins to be wrong. And depending on the kind of mutations and depending on which kind of protein is happening to, it can result in a malfunctioning regulator. Namely, I can have a green light that's always on. That's bad because now the cell will keep on progressing through the cell cycle without stopping, and the cell will divide, 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 divide. Or I can have a mutation that causes the red light to be permanently off. Without the red light, nothing is stopping the cell cycle. And again, it would just be keep on going and going and going and divide, 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 right? Do you understand those 2 points? One more time. These checkpoints are done by proteins, and they either speed it up or slow it down. It as in like the cell cycle. And if there are problems with these proteins because of DNA mutation, then the cell cycle will be going very, very fast. Okay? And we don't want cell cycle to be going very fast because then now you have uncontrolled cell division, which would result in a tumor. Okay? Having said that, let's take a closer look at the green light. So some genes, we call them proto-oncogenes. Proto-oncogenes. These are genes that would create the green light proteins that would help the cell grow. And here, I'll write down for you. I'll write it down for you in green. These creates green light proteins. Okay, I call them green light proteins, but they are called growth factors, right? Growth factors. Okay, so produce these green light proteins. However, if the proto-oncogene mutates Mutates means like changes. Okay? The DNA is wrong, it changes. Or sometimes you have too many copies of the proto-oncogenes, then the proto-oncogene has turned bad. It has become a bad gene. This bad gene might be permanently turned on, like the green light is always on, like I said, or permanently activated. When the green light is always on, then it's always go, 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 right? There's no stopping. The cell growth becomes out of control, which could lead to cancer. This bad gene, this one has the mistake. We call it an oncogene. What kind of doctor does a person go see when they have cancer? What do we call those doctors?
Oncologist.
That's right, oncologist, right? Yeah,
Oncologist.
yeah, oncologist, oncogene. They relate to the same thing. Are we together so far?
Yes.
Yes.
Okay, great. Now, the opposite of proto-oncogene is tumor suppressor gene. Okay, so tumor suppressor genes are genes that makes the red light protein. Okay, red light protein. Now, again, they're not really called red light protein. They are called growth inhibitors, right? Okay, but I'm calling them red light proteins to help you understand, visualize what's going on. So these red light proteins, they have many functions, but generally they slow down the cell division. Okay? When do you want to slow down cell division? Well, you want to slow down cell division if something's wrong at the checkpoint, right? If they have DNA mistakes, then you need to repair them. If you have errors that cannot be uh, 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 fixed, then you might want to kill the cell, right? Apoptosis. So these tumor suppressor genes will undergo these kind of functions. Okay, it will try to slow down cell division. It will help repair the DNA damage. It will kill the cell if necessary. All these actions aimed at one thing. They aim to prevent a cell from dividing. Okay, that's why they're called tumor suppressor gene. They suppress uncontrolled cell division, in other words. They are red lights. When the tumor suppressor genes, excuse me, as you can tell, I'm really sick, so I'm losing my voice. Uh, when tumor suppressor genes don't work properly, when the red lights are no longer functioning, then the cell can grow out of control, okay, which again can lead to cancer. So we can have get cancer problem in one of two ways. Either we have a green light that's always on, or we have a red light that doesn't turn on. Okay? Let me say that one more time. You should write it down. Write down. Green light, growth promoter, growth factor or green light, that's always on. That's bad. Or a growth inhibitor, a red light that does not turn on. Both situations can lead to cancer. Cancer is basically uncontrolled cell division, right? Now, exactly, it needs to be a balance of green light and red light. Okay. Now you might think, hey, what about a green light that doesn't turn on? Is that going to be a problem? Well, a green light that doesn't turn on is a problem, but it's not a problem that will cause cancer. Right? If you have a green light that doesn't turn on, then your cell doesn't really divide as quickly. So maybe you will have trouble healing the wounds. Okay, maybe you're not growing as fast. Okay, but it's not going to be cancer. Okay. Same thing. If you have a red light that's always on, so you're pressing the brakes all the time. Okay. It's not going to give you cancer necessarily, but it could cause other problems. Right. Like so we don't want the cell cycle to be too fast. We don't want it to be too slow. We want it to be just right. If you heard of the story of Goldilocks and the bears, right? You want it to be Goldilocks, just right in the middle. Not too fast, not too slow, the right speed. Okay? I feel like I'm doing an okay job so far. But here's the next part. All right. Now, what kind of problem will cause the cancer? I already told you it's a green light that's always on or a red light that doesn't turn on. But it's a little bit more complicated than that. It turns out for the proto-oncogene, we call these a gain of function mutation. What is gain of function? Basically, in short form, gain of function means always on, always active. Okay? The green light won't turn off. It turns out just one of these mutations is potentially enough to cause cancer. Okay? Think of a car. You have the accelerator to speed up, you have the brakes to slow down. It's kind of like the green light and the red light. But if you have a mutation in the accelerator and the accelerator is stuck, then the car is going to go really, really fast, right? And just one problem is enough for you to be out of control. And that's what happens here. You have two copies of the same gene, but if only one of them becomes permanently on, then we are going to be out of balance. Let me try to explain to you with these diagrams, okay? Just watch and listen and ask questions if you don't get it. So we have two genes, and one from mom, one from dad. Okay? Gene is like just a section of DNA, right? And there are like thousands of these genes. I'm talking about like a proto-oncogene, okay? So like we have two green lights here, and if they are on, then the cell will divide, but then they are not on forever. Sometimes they will be off, and then the cell don't divide. Okay, so divide, don't divide. And it's this balance of turning on and turning off that's going to allow the regulation of the cell cycle, right? Okay, never mind the red light right now. Okay, like it's really complicated, the whole thing. Right? I'm just trying to like simplify it down for us, right? So just focus on the green light right now. If green lights are on, then the cell divides. If green lights are off, then they don't divide. In a perfect world, if everything's working the way they should, both of them would turn on at the same time and both of them would turn off at the same time. But here's the thing. If I have one of these proteins, one of these DNA mutated, and let's say this one is busted now. Okay, the one on the left has a gain of function mutation, right? And it doesn't turn off. So when it's time to turn off the lights, rather than turning off both of them off, because the left one is busted, the left one has a gain of function mutation. I try to turn it off, only the right one will turn off, but then the left one will stay on. So when I'm a cell and I'm looking at the signal, I can see, oh, hey, the green signal is on. That means we can go and divide. I will follow the green signal. And then I will keep on dividing and dividing, even though you are not supposed to. We're supposed to turn it off. Because of the mutation, I can't turn it off. So you see, with just one mutation, that's enough to cause the cell cycle to go out of control. Okay, it's out of balance now. Any questions so far?
Can you explain this one more again?
Sure. Which part exactly?
Like light on,
The
like
whole
this.
thing? Yeah.
Yeah.
Sure. Okay. Do you understand when I say when both of them are on and the cells see these green signals, they will divide? Do you understand what that means?
Yeah, yeah, yeah.
Yeah. And then you turn them off, then they don't divide, right?
Mm-hmm.
Okay. Now here's the problem. These are proteins, and the proteins are made from the DNA, right? So if the DNA has a mutation, if DNA has a mistake that causes the protein to stay on all the time, and it happens to one of the two proteins, because we have two copies, right? But if one of them is broken, when I try to turn these lights off, only this one turns off. The left one will stay on. So from the cell's perspective, because there is still one green light, you're gonna follow the green light. You're gonna go and and divide, right? And it doesn't matter. You try to shut this thing off, you can shut it off because the green light is on, and the cell will just keep on dividing and dividing. Okay. I guess
Mm.
another way to think of it is think of
Oh,
driving,
so
right?
one gene,
Sure. Go ahead. Mhm. Yeah, go ahead.
So one gene get the instruction from other gene, like these are two
One,
separate gene.
oh, these are two separate genes, but of the same, because each of our gene has two copies, right? One copy from mom and one copy from dad, right?
Yeah, yeah. That's
Yes.
why if one is mutated, the other one gets signal from the which one is mutated. I mean, which one is green, so I can follow that one like this.
That's what the cell will not
No, no,
distinguish
not the
which
cell. I mean,
the gene.
the gene.
Okay, yeah.
Yeah.
So the gene they function
Or cell.
separately.
The whole cell
Uh,
cannot distinguish.
That's right. The
Whole
whole
cell
cell
cannot
cannot
distinguish.
distinguish. Yeah, that's
Yeah.
right. So
oh,
the whole cell is just
Okay.
gonna follow whichever one is on. Okay. And if even at least one of them is on, they're gonna go. Okay. I guess one way to
Yes,
think
yes.
about this is like when we're driving, there are two sets of traffic lights, right? Okay. If they are both turned on green, then we know we will go. But even if one of them is not functioning and then that one goes green, we know to go as well, right? So like we follow the one that's turning on. We don't look at the broken, the one that's turning off, right? Do you understand what I mean?
mhm.
Yeah. Any other question before it gets more confusing? It's going to be more confusing with a tumor suppressor. I don't know if this analogy is helping or it's making it worse.
So,
Uh,
so Professor, either one or two or both is green, then it has a possibility for cancer.
Exactly. So like if both is green or one of them is green, that's not really the problem. The problem is like if they're both green, that's fine. I divide. Under normal circumstances, both can turn off, right? So if it's off, then I'm not going to divide. If they're on, I'm going to divide. Normally, you can switch them off, switch them on, no problem. But it's when you have the gain of function mutation that causes one of them to be stuck on the on position, then it's always on, right? Like even the other one is turning off. It's like as if I can't see it. I'm just focusing on the one that's always on. And then I will go, go, go and divide and divide and divide and it becomes tumor.
uh, Teacher, as I understand, like they are working separately, like they has different functions. So if one of them follow uh, others, it's going to be wrong.
Another
Is
way
it
I
like?
can say, another way I can help you guys understand is when you have a signal that's stuck on the on position, it will
Mhm.
override the other one. It will
Okay.
cover up the other one. Okay. Um, and that's something we'll learn more in lecture 8. But basically in this situation, the malfunctioning light is overpowering the normal functioning one. Yeah, even though the normal one tells you to stop, we are going to ignore it because this one is the dominating one now. Okay, now this is going to make it worse. I'm so sorry, guys. Let me try my best here. The red light, they are the tumor suppressor gene. Tumor suppressor genes are supposed to slow down the cell cycle, right? So when the red lights are on, do I divide or not divide? I'm hoping you're
Not divide?
That's right. I'm hoping you're thinking not divide, right? So stop, not divide. If the red lights are off, then I can divide. Okay? So don't divide, divide. And again, it's this on and off that help me pace the rate of cell division. Now, for tumor suppressor gene, instead of a gain of function mutation, it is a loss of function mutation that's going to be problematic. In this case, instead of having a red light that's always on, we have a red light that does not turn on. Loss of function means it doesn't turn on. So what if I have one of the lights not turning on? Let's see what will happen. Okay, so this is both of them turning on, and then let's say the right one is broken. The right one has a mutation and it doesn't turn on anymore. Okay, so it doesn't turn on anymore. Am I still going to divide or am I not going to divide?
Divide.
Divide.
Okay, let's do it as a poll. I want everybody to think about this. Okay, there is only one red light. Think about driving, guys. If one of the traffic lights not working and then you see only one of them has a red light and the cop is in the corner, are you gonna go or are you gonna not go? Okay, that's what I'm trying to ask you to think about. Like, what's the safest thing to do? Do you go? Do you move or not move? If you only see one red light turning on, the other one is completely busted or like it's not turned on, do you go or do you not go? Right? Do you divide or do you not divide? So I haven't tell you which choice is which. I'm so sorry. A is for divide, B is for not divide. Do you divide or not divide? A is for divide, B is for not divide. Okay, yeah. So a lot of people, the answer's coming in. Exactly. You're not going to divide, right? You're not going to divide. I see the red light, right? Oh, we're not supposed to go. That one is not turning on, but it doesn't matter, right? I'm just gonna ignore. I'm just gonna follow this one, right? Okay. So you see, in this in this case, with the sorry, in this case, with the tumor suppressor gene, even if I have one copy that's not working, it's okay. It's not cancerous yet because I still have the other functional red light. The problem is going to happen when both of them has the loss of function mutation. In this case, when both of the red lights are not turning on, then there's nothing to stop me from moving. I don't see the red light turning on. I'm just going to go, right? And then it's going to divide and divide and divide and divide. That's why for the tumor suppressor gene, you need two mutations in the same cell to cause an uncontrolled cell division problem. Whereas for the green light, you only need one of them to be busted, and it's enough to cause the cell to have uncontrolled cell division. All right. Let me put it in words for you. Loss of function in tumor suppressor gene, both copies needs to be mutated. And then here it says people born with only one functional copy are more likely to get cancer. I'll explain this. I see someone has a question. Go ahead, Kadisa.
No, no, it's okay now. I clearly understand.
Okay, thank you, thank you.
Thank you.
All right. Now this last point is also difficult to explain. It's difficult to explain in virtual environment. That's what I mean. Um, but we'll see. Okay, there is another comment. So it's problem in both way when one is not working properly. Yeah, exactly, right? But you have to also think about what it means by not working properly. For the growth promoter, for the green light, it is always on that's causing a problem. Whereas for the tumor suppressor, it is always off that's causing a problem. Okay. So let's see. Let's say we have a bunch of cells. Okay, these are cells, okay? These are not green lights, red lights, these are cells. Each cell has two copies of each gene. Okay, two copies of tumor suppressor genes, two copies of proto-oncogenes. And I should tell you, there are multiple proto-oncogenes and multiple tumor suppressor genes in the actual cell. Okay, but for simplicity, we're just gonna focus on one set of tumor suppressor gene. Okay, so think about this. You don't have to answer me. Just think about it in your head. This is a healthy individual, no mutation whatsoever. But let's say just by chance, bad luck. Usually these things are bad luck. It's not things that you do or not do. It's just by chance it happens. Let's say this one, remember there are two genes, two copies of the tumor suppressor gene. Let's say one of them has a mutation. One of the red light is busted and it doesn't turn on. Is that enough to potentially cause cancer? Think about that. Is that enough to cause cancer?
No,
No, right? As I
no.
told you, we still have the other red light that's working, so it's fine, right? Now this one you really got to think about carefully. What about later on the person has another mutation, just again bad luck in this cell. Is that gonna cause cancer now?
Yes.
Yeah.
Sorry, I didn't understand.
So
Uh,
in this cell, one of the red light is not working. That doesn't cause cancer, right?
yeah.
I'm saying in another cell, what if there is a mutation now in tumor suppressor gene? So one of the red lights in this cell does not work. Now, is it going to cause cancer?
No,
No, it's not, right? Because look,
No.
this cell has
I
one
will go.
functioning red
Oh,
light.
that is another cell?
That yeah, this is a
Okay,
separate cell,
okay.
right? This one
Okay,
also has
yeah,
one
yeah,
functioning, right? Do you understand?
yeah.
So like, you can have a single mutation in these cells and it's gonna be okay. It's only if the same mutation happened twice in the same cell, then this one is now gonna be out of control, right? That one has no more red lights. That's working. And then it's gonna keep on dividing and dividing and dividing, right? Does that make sense? Now, we have trillions of cells in the body. What's the probability that you will have the same mutation twice in the same cell? It's not impossible because people do get cancer from these kind of situations. So it's not impossible, but it's low probability, relatively low probability. Okay. It's like hitting the same spot twice is unlikely event. Right? But what about people who inherited one broken red light from one of the parents? Remember, we get all our genes from mom and dad, right? What if they had a broken red light that get passed on to us? If that's the situation, all of the cells will have one broken copy to begin with. You see, all of them would have one broken copy of the red light. And now, if this individual has one mutation for the other red light in any of the cell, that's going to be a problem immediately, right? It doesn't matter if you have a mutation here, that's going to become cancerous. If you have a mutation here, that's also going to become cancerous, right? Now the likelihood is much higher because they inherited a non-functional copy of the gene from the parents.
That's why genetic doctor ask if you have any.
Exactly. That's why they ask, is there any familial history of cancer or anything like that, right? Not all cancer has genetic basis or um, they don't all have strong genetic connections, but a lot of them do. So that's what it means here. People born with only one functional copy are more likely to get cancer. Yes, another question?
No, but like a few months ago, I read something that was saying there was what can I explain? Uh, they
That's
are
okay.
search, yeah. Those people search about cancer, and they found out it's actually not genetic, and it's about your lifestyle. If your lifestyle is bad, you're gonna get cancer. But they were saying it's actually not genetic. So
Okay. Yeah,
that's why
so I
I
mean, we
confused.
we have to be very careful with what we read online. But there's certainly some truth to what you're saying. I can tell you definitively there are genetic components to cancer, but hold on to that thought. I have a slide coming up. Okay. Let's go back to this verse. Mutations could be inherited or acquired. Inherited, you are dealt a bad hand of cards from birth. You got the bad genes from the parents, and you have to live with it. Okay, but mutations can occur as you grow up. Okay, so how do you get mutations? Well, that's what you were saying, right? Is this Senna? Is that how you pronounce your name?
Yes, Senan.
Yeah, so as Santa was pointed out, right? It has to do with lifestyle. There are a lot of things that we do that would increase our risks of getting mutation. Example would be, UV light exposure. So that's why we have to put on sunblock, especially in the northern hemisphere where the sun can be very strong, right? We need to put on sunscreen to minimize UV exposure. Alcohol consumption, smoking cigarette, eating a lot of red meat, processed food, all these things will increase our risk of getting mutations, which could lead to cancer. So it's not 100% just your genes alone. The genes would increase your risk or lower your risk of getting cancer, but your lifestyle also plays a role, right? So there is a famous line in genetics that's called um, nature versus nurture. Nature is what you're born with, your genetic stuff. Nurture is your environmental exposure. Okay, two twins that are born genetically identical, if they grow up in different environments, they could look very different, right? Because of the nurturing factor that causes the manifestation of their genes to be different. Okay, so back to our point here. When someone has inherited an abnormal copies of a gene, their cell already start out with one mutation, and it makes it easier and quicker for enough mutations to build up for a cell to become cancer. Now, once again, I have to emphasize, I am simplifying these things, okay? It's super, super rare for a single mutation to cause cancer right away. It's usually like a bunch of mutations. So like a bunch of green lights that are permanently stuck on and a bunch of red lights that are not turning on, then it leads to cancer, okay? But like I'm oversimplifying things for us to understand it. That is why cancers that are inherited, so you inherit one copy, tends to occur earlier in life rather than cancer of the same type that are not inherited. Okay. If it's not inherited, you have two chances. Okay. And the chance of the longer you live, the higher the chance you get the same mutation twice in the same cell. Okay? But if you're dealt with one bad copies, it could happen way earlier in life. And there is also this concept of penetrance. I want you to remember this word, penetrance. Penetrance, in a nutshell, refers to how likely the gene will actually, sorry, let me try that again. Penetrance refers to how likely a certain mutation would actually translate to a disease. Okay, like if I inherited this cancer gene, like, am I absolutely going to get cancer? If that's the case, we call it complete penetrance, 100%. Incomplete penetrance is not 100%. Okay? You have a higher risk of getting it, maybe 50% chance, okay, or like 20% chance. The lower the penetrance, the less likely this gene, this mutated gene, is going to cause a problem. Okay, so just because you get a bad mutation is not automatically going to be a cancerous situation. Okay, that's why like a lot of people like they do genetic there are companies that allows you to send in your saliva and then they give you your DNA profile and tells you what kind of mutations you have, and they would actually tell you how long you're gonna live either, right? So a lot of those are like, not good science, okay? Like because of things like penetrance. Okay, we shouldn't, in my opinion, we shouldn't be believing all those things. Anybody who can tell you how long you live exactly, you should be skeptical about them. Okay. All right. Now there is something called the BRCA1 gene. Okay. BRCA1 gene stands for breast cancer 1 gene. It is an example of a tumor suppressor gene that plays a role in repairing damaged DNA, destroying cells if you cannot repair the DNA, and therefore it prevents the cell from going into cell division. So this is a tumor suppressor gene. So BRCA1 gene is one of the contributing mutations that would lead to breast cancer. In general, 12% of women in the population will develop breast cancer sometime during their lives. Okay, 12%. However, up to 55 to 65% of women depending on the demographics and where they live and ethnicity and all those stuff, 55 to 65% of women who inherited one harmful copy of BRCA1 mutation will develop breast cancer by age 70. So this is what I mean by whether you are born with no broken red lights, or if you're born with one broken red lights. If you inherit one broken red lights from the parents, the risk of getting the same type of cancer increase by 5, 6 times, right? It's only 12% in the general public, but if you have one broken red light, the chance is much, much higher because you only take one additional mutations to kind of push the cell over the edge. Do you understand? Some of you know who she is, right? She's an actress, Angelina Jolie. Her mother died of cancer, and I think her sister did as well. So she went and screened her DNA profile, and she found out she inherited the mutated BRCA1 gene. Okay, I want to clarify. Everybody has the BRCA1 gene. Male has it, female has it. Okay, it's a red light protein gene. It's a gene that makes red light protein, okay? But we're talking about like a mutated version of it. So she inherited a mutation copy from her mother, which increases her risk of getting breast cancer, right? As I told you already. So as a prevention, she actually underwent double mastectomy. Right? She basically removed her breast, and then she had reconstructive surgeries to uh, to recreate the breast, and that basically would remove her risk or lower her risk of getting breast cancer substantially, right? You can read more about it. She did an interview on this. I'd love to tell you more, but we don't have time. We have to keep going. Okay, so cancer is uncontrolled cell division, and this uncontrolled cell division, this accelerated uncontrolled cell division, result in the formation of a tumor. Okay? Tumor, also called neoplasm, a new blob of cells. That's what neo means. Neo is new, right? So the tumor is not necessarily cancerous. If someone discovers a tumor, first thing they have to do is they have to do a biopsy. Right? They have to take a sample of the tumor and see whether it is cancerous or not. If it's not cancerous, we call them benign. Right? They're not dangerous. Sometimes you can surgically remove it and it wouldn't cause any problems, and they don't come back. Right? But if it's cancerous, we call them malignant, malignant neoplasm. Right? That's the cancerous kind. That's the kind that nobody wants to have. All right. Benign is okay, malignant is not. So how do you know if something is malignant or if it's benign? Well, there are 6 hallmarks or characteristics that are used to define whether something is cancerous or not. I'm going to quickly show you what they are. Number 1, cancer cells have self-sufficiency in growth signal. Because they have their own green light that's always on, they don't need other signals to tell them to grow. Okay, if you like in a normal cell, you need to turn on the green light for the cell to divide, right? But they don't care. They have their own green light. You don't need to tell me to go. I'm just gonna go. Okay? Number 2, insensitivity to anti-growth signals. Okay, you tell them to stop, they don't stop. Right? They ignore the red lights. Okay, they just keep on going. Right? It doesn't matter. You tell them to stop growing, you give them signals to tell them to stop. They are insensitive to it. Okay? Insensitivity to anti-growth signal. It's like my kids. Right? Doesn't matter how many times I tell them stop fighting, stop hitting each other. They ignore me. They're insensitized to my anti-fighting signal. Okay, so self-sufficiency to grow, insensitivity to anti-growth. That's the first 2. Next, they can evade apoptosis, evade death. Okay, so a Grim Reaper comes and try to kill it, it doesn't care. It's not gonna die. Okay. Sustained angiogenesis. Okay, so angiogenesis means formation, formation of formation of new blood vessels. To sustain all these growth, because remember the cancers keep on dividing and dividing, you need a lot of resources and energy and ATP and oxygen and all that stuff. It needs to establish its own line of supply. So it can promote formation of new blood vessels that would leach nutrients and energies from surrounding cells and tissues to support the growth of the tumor. Okay. And that's basically what cancer is. Cancer, it sucks the life out of people by taking up all the resources for healthy cells, and the body wastes away. Next one, limitless replicative potential. Remember, the telomeres, once they get really, really short, they are supposed to stop the cell from dividing, right? They might trigger senescence, right? They will cause the cell to become old, but not for cancer. Cancer, they found a way to turn on telomerase. So it gives them really, really long telomeres. And then they could divide and divide without having to worry about becoming senescent. And finally, when they grow so much, when they have tapped out the resources of the original spot, then the final stage is they could spread and invade to other tissues. That's called metastasis. Okay, and like this is the worst. Once it has spread to other organs, it's almost point of no return. Right? There's no more cure for it. All right, so those are the 6 hallmarks of cancers. Okay, just one more time quickly. They can grow on their own with their own green lights, with their own growth signals. They are insensitive to red lights. You tell them to stop, they don't stop. You tell them to die, they don't die. Okay, evade apoptosis. They could create their own supply of nutrients. They could keep on dividing and dividing with no stopping because of the long telomeres. And once they use up all the resources, they can invade and spread to other organs. Okay, and that's it. I mean, there are a couple of questions here, but I'll post the answers and you can check them out. Um, Next, yeah, we still have 2 more classes to do lecture 7 before the quiz. If we don't finish, I can move it, not a problem. But on when wait, is this already Wednesday today? No, today's only Monday. I'm so sorry. It's Wednesday. We'll do lecture 7, and can you please print something for lecture 7, because like you need to have this thing. It's much easier if you have a physical copy of it to do it, okay? Just hold on a second, I'll show you and I'll let you go. Protein synthesis. Obviously, printing the notes is good. It's important, but even more important, I want you to print the codon chart here. Okay, codon chart. This is like the periodic table I was telling you about. Okay, print a copy, and then I'll teach you how to use it next class. Thank you, guys. Take care. See you Wednesday. Bye-bye.
See you. Take care.
Thank you.